Hello students, today I shall take up the lecture 7 of the first chapter Herbs as Raw Material, that is Ayurvedic preparation, from the subject Herbal Drug Technology, code 603T. I am Dr. Kumud Madan, Associate Professor, Lloyd Institute of Management and Technology. Students, in the previous slides, we have studied about the various principles of the Ayurveda, what Ayurveda is based upon. Now, we have to study the different dosage forms which are available in the Ayurveda. So, Ayurvedic dosage forms, they have been divided into the different categories depending upon their physical nature. These categories are solid dosage forms, which includes Vati or Gutika, semi-solid dosage form which is called as Kalk or Avaleha, liquid dosage form which includes Arishta, Asava ya Tel, powder dosage form which includes Churna and Bhasma. We need to standardize the different Ayurvedic dosage forms because they are obtained from the minerals, and certain minerals are not desired to the extent they are involved. Hence, there is a need of standardization as asked by the scientific community of the world. Ayurvedic preparations are polyherbal or herbo-mineral preparations. Even if a single herb is used in the preparation, the single herb will contain multiple constituents. And if we talk about the modern science, we are targeting a single phytoconstituent for a particular disease. We feel there is no need to add additional constituents or the agents which are not required. But the Ayurvedic preparation contain more than one elements, more than one components and more than one minerals. Hence, there is a need to standardize these. Bioactive chemical constituents are not known in the herbal preparation, and even if it is known, it is compared with the markers, it does not necessarily reflect its connection with the biological effects. Although we have been using the Ayurvedic dosage forms in our homes since time immemorial, but now the question is being asked by the scientific community that why there is need of adding so many components. What is the role of each and individual component? Say for example, अगर आप किसी भी प्लांट की एक्सट्रैक्शन करते हैं, say for example, अगर आप ट्राइकोस्पर्मम एमी अर्थात अजवाइन का सीड का एक्सट्रैक्शन करते हैं, you get more than 100 components. But only few components have their established therapeutic or the biological action. So, the question comes is, what is the use of the other components? Why we are not taking just one phytoconstituent? Why we are not isolating just one phytoconstituent after the extraction and using it? So, there is a need of the standardization of these Ayurvedic dosage forms. That is based on the principle of holistic approach does not permit assaying a single marker. Now let us study the different Ayurvedic dosage forms and their standardization or the quality control parameters which has to be taken care of. Asava and Arishta. These are liquid preparation or self-generated herbal fermentation products. Asava are prepared by direct use of fresh herbal juices or herbs soaked in water. Arishta are prepared with the decoction of the herbs in the boiling water. Both of them have sweet taste and alcoholic smell. Now, what are the steps in preparation of these? Powder herbs are soaked in herbal juice or decoction in presence of sugar or jaggery for specific period of time, fermentation is brought by the addition of the dhatuki dhai ke phool, which is having the chemical name as Woodfordia fruticosa flowers. This fermented alcohol, what it does here, it helps in the extraction of active constituents in the herbs, and it also acts as a preservative. So, these preparations, they generally contain 12% alcohol, and hence they are also known as medicinal wines. Now, the standardization of Asava and Arishta. They should be clear liquid without any froth. Froth is a symbol of the fermentation, which should not be present in the final preparation. They have pleasant aromatic odor of alcohol, which they contain around 12%. They should not have a sour taste. The fermentation should be carried to a specific time. It should not go beyond that, giving the sour taste. That is, they should not have the khatta taste. They should not be additive sink to the bottom. They should not settle down. It should be uniform in the dispersion. Burning candle burns brightly when placed in or just above they are placed in the sandhan patra, that is the vessel which there is used for their storage. Besides that, organoleptic evaluation of these preparations needs to be carried out. The first one includes physical evaluation, which checks the pH and the specific gravity and 25 degrees C and the total solid content. Besides that, it also goes for the alcoholic content of the preparation. This is the test for the reducing sugar as well as non-reducing sugar should be carried out. The test for presence of methanol, acidity should be carried out. They must undergo the process of chromatography, and they must be checked for their shelf life as well as viscosity. The next preparation which we shall discuss is Churna or Churnas. Churnas means the powder dosage form. Normally we take Churna. These are solid Ayurvedic preparation of the powder of drugs, which often taken with some anupan. Anupan means the substance which is taken along, which can be milk, ghee or honey. चूरन को जनरली घी, शहद या दूध के साथ लिया जाता है. शहद के अंदर उतना चूरन जो आपकी डोज है उसको डाल के इट इज़ मेड एज अ पेस्ट एंड देन इट इज़ कंज्यूम्ड. एंड सिमिलरली इट कैन बी टेकन विद द घी and it can be taken with the milk. You can have the churn and then you can take it inside your body with the help of a milk as a liquid. These should be fine at least of the 80 mesh size. So, when the churns are made, they are undergoing the sieve analysis to ensure their size. It should be kept in the airtight container so that there is no entry of the moisture, because moisture is going to promote the growth of the microorganisms. It should be used within 6 months. So, that is general shelf life of these churnas. How they are prepared? Now, let us study the preparation method. Make powder of crude drug and sieve through cloth, which is generally taken as a muslin cloth or a fine cloth or mechanical sifter. If there is more than one drug, each drug has to be separately powdered and then mixed. Besides that, care is taken that if the presence of salt, sugar or camphor is there mentioned in the formula of the churn, that has to be size reduced in a different vessel and then has to be mixed. So, if the formula of the churn contain 4 drugs, all individual 4 drugs has to be size comminuted or the size reduced or turned into powder separately, and finally they have to be mixed. Now, the standardization of the churn includes the study of its organoleptic characteristics. This include color, odor and taste. How does it look like of the color? Odor, does it have a smell or a fragrance? Or what is the taste of it? Similarly, the determination of physicochemical properties is also done, which includes the taste such as moisture content, total ash, acid insoluble ash, water soluble ash, water soluble extractive content, alcohol soluble extractive content and crude fiber content. So, these are the different quality control parameters which have to be carried out for the standardization of the churnas. Evaluation of the churnas also includes the quantitative estimation of the selective phytoconstituent, which is done by the technique which is specific for a particular phytoconstituent. Evaluation also includes the powder fineness, bulk density, tap density, angle of repose, compressibility, Hausner ratio, determination of the pH, establishment of the safety pertaining to heavy metals and microbial load. These are the generally tests which we carry out for the any powder dosage form in the case of allopathy as well. We often perform these tests for the granules as well. Bhasma. Bhasma is solid Ayurvedic preparation which contains metals, minerals and animal products which are burnt to ash. They are generally white, pale yellow, black or red in color. Organoleptic characteristics of Bhasma include sound. This is imperceptible sound on grinding the Bhasma between the teeth. When it is ground between the teeth, there is a sound which shouldn't be audible enough. Touch. How do they feel when you touch it? So, they should be soft or imperceptible coarseness in the Bhasma should be felt. Taste. They should be tasteless. Odor. They should be odorless. Besides that, the other physicochemical parameters of the Bhasma include pH, ash value, acid insoluble ash, particle size distribution, X-ray diffraction, inducively coupled plasma emission spectroscopy, UV vesicle spectrophotometry. These are the analytical techniques which establish its physicochemical characteristics and hence help in the standardization by doing the quality control. Next preparation is the semi-solid preparation which is called as Avaleha. These are semi-solid preparation of the drug prepared with the addition of the jaggery, that is gur, sugar or sugar candy and boiled with prescribed drug juice or decoction. A very well known common example which exists in our homes, that is Chyawanprash. Chyawanprash is an example of the Avaleha which is semi-solid in nature. Yes, we all can make it, that it is semi-solid in nature and it consists of the different drugs which are basically prescribed with the some sweet tasting as well. Chyawanprash में मिठास भी होती है. Avaleha are also known as modak, gud, khand, rasayan, leha because they are sweet in nature. The preparation generally have kasai or other liquid. Now, this is the composition of the Avaleha. It consists of jaggery, sugar or sugar candy. It consists of powders or pulps of certain drugs. It also consists of ghee or oil and honey. Now, let us study the characteristics of Avaleha. Avaleha should neither be hard nor be thick fluid. When pulp of the drug is added and ghee or oil is present there, the preparation does easily roll in the fingers. So that is the final sign of its quality. The growth of fungus or the fermentation, if present, is a sign of deterioration. So such Avaleha should be prohibited, and we should make ensure that the growth of microbial attack doesn't happen to be. Whenever there are metals which are mentioned in the formula of the Avaleha, so they are not used directly. Instead, the Bhasma or the fine character form of the metals is to be used. In the case of drugs like Bhalatka, purified drug alone are included in the preparation. Besides, the color and the smell of the Avaleha depends upon the composition or the drugs which are used in its preparation. Preservation. The Avaleha or leha should be kept in glass or porcelain jar. It can also be kept in a metal container which does not react with it. Normally, they can be consumed within a year, or this is, say, its shelf life. Next preparation we need to study is Vati and Gutika. In the earlier classification we studied these are the solid dosage forms. Vati, you can say, a sort of a tablet. So, these are the medicines which are prepared in the form of the tablet or pills. They are made from one or the more drugs of the plant, animal or mineral origin. The preparation method include the following steps. Number 1, the drugs which are mentioned in the formula, they are converted into the fine powder. Solid drug को हम size reduction करते हैं, powder में reduce करते हैं, and then the minerals are made into bhasma. जो भी mineral elements present हैं उनको हम भस्मिक में convert करते हैं, भस्म और सिंदूर. Unless otherwise mentioned, in cases where parad and gandhak are mentioned, parad and gandhak are basically related to the mercury of which toxicity has been reduced. So, whenever they are mentioned in the formula, kajli is made first. Kajli is a process by which the mercury is converted into intoxicated form. We all know that mercury is a sort of a poison or the toxin. So, kajli is made, that is a preparation where the toxicity of the mercury is decreased or lessened, and this kajli is further made and the drugs are added one by one according to the formula. So, this is the way a vati is prepared. I am going to just summarize. Whatever drugs are present, they are converted into solid, from the solid to the powder form. If metals are present, they are converted into the bhasma form, and then kajli is added. If the parad and gandhak are added, they are converted into kajli before adding. Now, what is done? These are put into khalv. Khalv is a sort of a pestle mortar. It is an oblong pestle which you might have seen in the advertisement of the Ayurvedic dosage form. These are put into the khalv and ground into a soft paste with the prescribed fluid. This is a process of trituration, if we call in the normal language. The mass is properly ground and is in a condition to be made into pills. This is triturated and finally converted into the pills, which is vati. The final stage of the formulation before making pills is that it should not stick to the fingers when rolled. So, this is similarly what we do at the stage of the granule making in the tablet. We generally take the mass of the coherent drug along with the adsorbent and we just press between our thumb and see the impression. Similarly, in the Ayurveda also, when vati is supposed to be made finally in the pill form, it has to be seen with this physical test that it should not stick to the fingers when rolled. Then pills may be dried in the shade or in the sun as specified in its literature. Now come to the standardization of Vati and Gutika. They are standardized or they are going under the quality control test as mentioned here. They have to be checked for their particle size. Identification is done with the techniques of microscopy, HPLC, TLC. Test for heavy metals and toxin is to be done. Test for microbial contamination if any is to be done. There is a check done for the presence of the pesticide residue. Physical evaluation of the Vati includes ash value, acid insoluble ash, water insoluble ash, alcohol soluble extractive, pH, volatile oil, melting temperature, loss on drying. All these tests are given in the Ayurvedic Pharmacopoeia of India and they are done accordingly as mentioned in the official monographs of a drug as well as its dosage form. These are the analytical techniques which are to be done for the analysis and the final quality control of the Vati and Gutika. So, here we end with the different Ayurvedic preparation, their standardization. Thank you.